Assalamu alaikum students. Hope so you all will be fine. Students, in this video I will discuss about the HDL metabolism. Okay, we will cover this topic under the heading of the site for the metabolism of HDL, then its maturation, the steps for the metabolism of HDL, and uptake. Okay students, so let's start. There are two sites for the metabolism. There are two sites where HDL gets synthesized. One is liver and the another is intestine. This is the liver, this is the intestine. Students, this is the liver, this is the intestine. These are the two sites where HDL gets synthesized as a nascent HDL. But here the shape of HDL is a disc like. Here it is a disc like shape of a nascent discoidal HDL. This is a phospholipid. Here is a free cholesterol. It also contains ApoC2 protein and ApoE protein. It is a nascent discoidal HDL. Students, intestine acquires ApoC2 protein and ApoE protein from liver. Here is also ApoA protein. Friends, I will repeat it again. The synthesis of HDL takes place in liver and intestine as a nascent discoidal HDL. Intestine acquires ApoC2 protein and ApoE protein from liver. This is the phospholipid. This is the phospholipid. This is the cholesterol and A1 protein, and this is proteins. Nascent discoidal HDL. Later on, here this nascent discoidal HDL gets secreted in plasma. This nascent discoidal HDL, it gets secreted in the plasma, and where it gets attached with lecithin cholesterol acyltransferase enzyme. Lecithin cholesterol acyltransferase enzyme. Now here peripheral tissues, there is a there is a transfer of there is a transfer of cholesterol from peripheral tissues. Transfer of cholesterol from peripheral tissues. But this process happens with the help of an important protein, and its name is Its name is ATP binding cassette transporter protein. There is a transfer of cholesterol from peripheral tissues, but this process happens with the help of an important protein, and that is ATP binding cassette transporter protein. Okay, students. Now here there is a role of lecithin cholesterol acetyltransferase enzyme. It is very important for the esterification of cholesterol, because cholesterol it gets attached with the nascent discoidal HDL, and here the esterification of cholesterol occurs. How? Let's see. Here is a phospholipid lecithin, the fatty acid, mainly unsaturated fatty acid, which is attached to the carbon number 2 of this lecithin, and this fatty acid transports toward the third position of cholesterol, and it converts into with the help of enzyme lecithin cholesterol acetyltransferase, it converts into cholesterol ester and lysolecithin. Okay, students, in this way lecithin cholesterol acetyltransferase enzyme performs work. Here is a transfer of cholesterol with the help of ATP binding cassette transporter protein, and this cholesterol gets attached to this nascent discoidal HDL, and this lecithin cholesterol acetyltransferase enzyme, it is responsible for the esterification of cholesterol. But basically what happens, the unsaturated fatty acid which is attached to the carbon number 2 of lecithin, it transports toward the third position of cholesterol and converts into cholesterol ester and lysolecithin. This is the function of lecithin cholesterol acetyltransferase enzyme. Now, and cholesterol ester, this cholesterol ester being hydrophobic, it gets attached to the inner part of nascent discoidal HDL, and this shape converts into the spherical shape. Here, this is the spherical shape of HDL. This is a cholesterol ester and ApoA1, ApoC2 protein, ApoE protein. This is called as HDL3. Now, in next case, there is a transfer of this cholesterol ester to the various other lipoprotein in response to triacylglycerol, and this process happens with the help of cholesterol ester transfer protein. Cholesterol ester transfer protein. This with the help of these proteins, this cholesterol ester get transported toward the VLDL and LDL, and here from this lipoprotein triacylglycerol it gets transported to the HDL3, and this converts into HDL2, which contains high amount of triacylglycerol and less amount of cholesterol ester. This is the HDL2. The main difference in between HDL3 and HDL2 is that the HDL2 contains high amount of triacylglycerol as compared to HDL3. This cholesterol ester transfer protein, it is responsible for the transfer of cholesterol ester to these lipoprotein, and here this lipoprotein transfer triacylglycerol to this HDL3 and helps in the formation of HDL2. Okay, students. Now HDL2 it is uptake by liver. It is uptake by liver with the help of some proteins, with the help of some receptors, and these receptors are HDL receptor, and these HDL receptor belong to scavenger receptor B1 family. With these receptors, this HDL is taken up into the liver, and where this cholesterol ester it converts into the either bile formation or bile acid for further metabolism process. So here you can see that why it is called as HDL called good cholesterol, because it is responsible for the transport of this cholesterol, reverse transport of cholesterol from peripheral tissue, this liver with the help of HDL. Reverse transport of cholesterol from peripheral tissues to the liver. Okay, students, I'm repeating again. This was all about the HDL metabolism. Basically, the HDL gets synthesized in liver and intestine. There is a phospholipid, free cholesterol, ApoA1 protein, ApoC2 protein, and ApoE protein. And this is called a nascent immature discoidal HDL. Here, disc-like shape. Disc-like shape. In next step, this gets secreted in plasma, and when it gets attached to this enzyme, lecithin cholesterol transferase enzyme, and here, the peripheral tissues, there is a transfer of cholesterol with the help of ATP binding cassette transporter protein, and cholesterol gets attached to this nascent discoidal HDL. Lecithin cholesterol acid transfer enzyme is responsible for the esterification of this cholesterol and converts it into cholesterol ester and lysolecithin. Cholesterol ester, being hydrophobic, gets attached to the inner part of this nascent discoid HDL and converts it into the spherical shape of HDL, which is called as HDL3. Okay, students, in next step, this cholesterol ester get transferred to the various other lipoprotein with the help of cholesterol ester transfer protein, and in response to triacylglycerol. In response to triacylglycerol, here is the HDL2 formation, which is uptaken by these receptors, HDL receptor that belongs to the scavenger receptor B1 family. And here cholesterol ester converts into the bile and bile acid for further metabolism process. So students, this was all about the HDL metabolism. Keep watching and thank you.